looking at the vegetative structures of fungi. Okay, so we have unicellular types and we have filamentous types. Okay, so there are fungi which are single celled. Okay, this one single celled. These are your you have, some of you love to bake, no? Some of you like to bake, you bake breads. I'm talking about bread, okay, not cake, but some of you like to bake bread. So what do? You took a bowl, no? Then you added flour, no? Then you add salt, right? Then you added yeast, no? Y-E-A-S-T. You added yeast, right? Yeast is nothing but your Saccharomyces, okay? That is the scientific name of yeast, okay? Locally, we say, like a common person will say yeast, nah? But you should know, okay? You are scientist, you are botanist, okay? Yeast is Saccharomyces, and the yeast, no? They are fungus, but they are single cell, okay? Like this. Let's say this is the nucleus, no? Like that, something like this, they have that sort of shape. Okay, they are not filamentous, they don't have branch. Okay, they are yeast. If you see, no, obviously you cannot see, right? But if you could see through the microscope, or just Google, just Google the structure of yeast, no, in the microscope, you'll see that they are single cell. Okay, so now you have learned something. Okay, something that you do every day, something you eat every day, no? Bread and butter we eat every day, right? Or bun, right? Or what to say, the sweet bun or the plain buns or all those, okay, all the baking that requires yeast is done with your Saccharomyces fungus. Okay, so that is your yeast. Okay, and they undergo fermentation, right? They grow and expand and make your bread nice and fluffy. Okay. Okay, let's read what they have written. Except certain unicellular forms, that is like Syncytium or Saccharomyces, the fungus are generally thalloid and filamentous type. Okay, thalloid means like your hand, no? It's like flat, okay, like something like flat. Do you remember when you sliced your bryophyte? Remember? It's like a duck foot, no? Like it looks like a duck, like the foot of a duck. It's flat, like it's flat. So that is what we mean by the word thalloid. Okay, like a thallus. Thallus means like a flat, like your hand, no? Like that, like a foot or a hand. Okay. Okay, so the fungus are mostly thalloid and filamentous type. Okay, so thalloid, because you cannot make out whether it's a root, a stem, a leaf, a branch, an arm, a neck, ear. Okay, it is just like a mass. Okay, so the filaments are known as hyphae. Okay, so if I draw, suppose this is some filamentous fungus. Okay, like that. So these filaments, okay, these are each cell wall. So these filaments, they are known as hyphae. Okay, let's go down. So when these hyphae, they get tangled up, you know, like they grow so much and they're so tangled up, no, you cannot make out which one is which. That tangled up mass is known as your mycelium. Okay, so let's repeat again. They may be unicellular, like your yeast, okay, Saccharomyces. They may be filamentous, okay, and the filaments are known as hyphae, and that mass is known as mycelium. Okay, so I think that part is clear. So that hyphae, it may be aseptate, okay, which contains many nuclei and vacuolated cytoplasm. See what this line means? This line, when they say aseptate, it means, let's say this is a fungus and these are the hyphae. Okay. Usually what happens, there should be some cell wall, no? Cell wall somewhere. But here, no. Remove that cell wall, remove the cell wall. I'm removing, I removed the whole thing. Okay. Okay. So there are no cell walls, but what happened is, okay. suppose this is a cell that is undergoing mitosis. Okay, so what happened? The chromosome will replicate, it will duplicate, right? And then it will form the cell wall, no? Then your in telophase, one will go to one pole, another go another pole, and then you get two cells, right? Two separate cells with a nucleus in between. But what happens if, fine, this was the, let's say the DNA, okay? Then it doubled, okay? It doubled, okay? That DNA doubled. Then it's separated. But instead of forming this wall, the wall never formed. So we are getting two nucleus inside the same cytoplasm, aren't we? Inside the same cell. So this happens when the hyphae are aseptate, means they don't have a wall to separate the nuclei, and then so many nucleus. I'm just using my finger to draw, okay? So please bear with me, okay? It's not perfect, the drawing. Okay, so you will have so many nucleus. This is 1, 2, 3, 4, 5, 6, 7, 8, 9, should be even, no? 10, okay? So all the nucleus are inside the same cytoplasm. Okay, so maybe aseptate and contain many nucleus and vacuolated cytoplasm, okay? The vacuole is like an empty space, right? But it's not like a vacuum, not like in space. That vacuole, it contains some sort of waste material, some sort of minerals, okay, all the waste material. So just because it's called vacuole doesn't mean it's a vacuum, doesn't mean you're in space where there's nothing, no. It has something, but not like the important things. Okay, let's see again. So when the hypha has so many nuclei, okay, because there was no cell wall formation, we will call it as a cenocyte, okay. The multinucleate tubular aseptate hypha is called as cenocyte, okay. In fact, any cell, okay, let's say this as a plant cell, it has so many nucleus, okay, in its same cell wall, same cytoplasm, we'll call it a cenocytic plant cell or cenocytic bacteria, anything which has so many nuclei without a wall is your cenocytic, okay. So here also no exception, multinucleate will be called as a cenocyte hypha. Okay, then the hypha which has partition walls dividing the hypha into many cells is called as septate, okay. So a septum is a wall, right? So if it has a wall, it's called a septate. If it doesn't have a wall, it's a septate, okay. Just like your symptoms, no? Some COVID patient, they show symptoms. They cough, they have dry cough, they have fever and all, no? So those are symptomatic. But some, they are walking around like nothing happened. So those are asymptomatic, okay. They are carrying the virus and they are super spreading to other people because they don't know, they don't feel it, no. So same thing, aseptate and septate. Something which doesn't exist is with the word a, okay, aseptate or asymptomatic. Okay, let's continue. I overspeak. I get it. I overspeak and the time just runs. Okay, let's continue. The septate mycelium in its cell may contain only 1, 2 or many nuclei with vacuolated protoplasm. Okay, now this sentence, see what it's saying is that in a septate mycelium means in a mycelium where cell wall is there, sometimes there may be 1 nucleus, sometimes maybe 2, okay, sometimes maybe more, okay. So because you must be thinking, this miss, sometimes she says septate, sometimes she say aseptate. No, it's not that. It's because fungus, these are lowermost organisms, okay. They were there, like I told you, no, they were there before us, long before us. They'll be there long after us, you know. So they are not so highly evolved like me and you. Me, I have 1 cell, 1 nucleus. Same with you. You have 1 cell, 1 nucleus, 1 nuclear membrane. You have 23 pairs of DNA. Fine. our cells are so we are highly developed organism, no? But these fungus, they're not highly developed. The septate, right? The wall is forming randomly, like blindly, no? You just form one wall to the left, one wall to the right, without any proper planning. Okay, so for example, in this mycelium, let's say the nucleus has been dividing a lot. Suddenly it'll decide, I've enough divided mycelium, I've enough divided my nucleus, just form wall. Sometimes it'll fall here, sometimes it'll fall there, sometimes it'll form here. Like it's random. So that's why you may have in one cell wall of the nucleus, I mean of the cytoplasm, you may have one nuclei or two or many. Okay. So what about this word, protoplasm? I'm sure you have heard about the word protoplasm in school, but I will take it that might be some of you don't know. It's better to be revised. Even if you know, what's the harm in revising, no? Okay. So, you know nucleus? You will say yes, miss. You know cytoplasm? I'll just write cyto. Okay, when the nucleus and the cytoplasm, they exist together without a nuclear membrane, okay, or without a membrane between them, we will call it joined as a protoplasm. Okay, let me repeat. Okay, the nucleus is usually inside a nuclear membrane, right? Then the cytoplasm is inside its another membrane. But together, when they exist together, okay, we'll call it as a protoplasm. Okay, when we want to refer to both the cytoplasm and the nucleoplasm, means it's the same matrix which is your cytoplasm is in the nucleoplasm, is in the nucleus, no? So that we will call it as our protoplasm. Okay, I hope it's clear. Nucleus plus cytoplasm is equal to protoplasm. Okay, very good. Then let's continue. The septa between the cells may have different types of pores. Okay, in geotrichum, small pores are present on the wall called as micropore. Okay, so these cell walls, they will definitely have to have some pores, no? Like us, we have tube. Okay, me and you, we have tubes. These tubes are what? Veins and arteries, no? In plants, they have what? They have tubes also. Those tubes are what? Xylem, phloem, no? Which transport water, mineral, whatnot, no? And these lower organisms like fungus, they will simply have a pore, okay? That will send the cytoplasm from one cell to another, transporting food and minerals, etc. Okay, let's continue. When, what is the next line? When only one bigger size pore is present, it is called as a simple pore. Okay, when there's a big pore between one cell to the next, this is known as simple pore. It is found in Ascomycotina and Deuteromycotina. These are families of fungus. Okay, like your plant kingdom, no, you have one family is bryophyte, another family was your pteridophyte, then another family was your algae, remember? So even these fungus, they have different families, okay, belonging to different weather depending on their structure or their composition, etc. Okay, if one group of fungus is similar with another group, they are in one family. Okay, like me, my father, my brother, we are all similar looking, so I'm in one family, right? Okay. How do I close this now? Okay. Then in other fungus like mucorales, trichomycetes. If you remember, girls, in the practical video, no, I sent. One was where we measured the size of a spore, remember? And I showed you the measurement of the mucor spore. These mucor, no, they are very large. Oh, again. Like these mucors, which belong to the mucorales family, they are very large, okay, very large fungus. When I say large, doesn't mean like football large, but when you look through the microscope, they are comparatively large, okay. The smallest will be, you have guessed it, the yeast, single cell one. So these are quite large, okay, as compared. So in the mucorales, the pores is surrounded by an overarching bifurcation of the marginal partition wall, which looks like a boarded pit of trachea. Okay, so let's look at the boarded pit. These are the micropore. Okay, remember in geotrichum, small pores. Then if there is a single pore, right, we call it as a simple pore. Then where is the where is that trachea, boarded pit? This one, okay. 1 minute. Okay, so these are your border region, okay. These are the border areas, okay. So in the next semester, you'll be learning about xylem vessels, tracheids, xylem elements and all, no? So they are almost similar to that, okay. What is this? Biumbonate, electron dense plug of material is present between the two bifurcations, okay. This one, this is electron dense. When we say electron dense, means when you look through the electron microscope, the light cannot pass through, okay. So it appears as like if you do an x-ray, no, you can see your bones, right? Your bones, because they are dense. So same thing, but when the electrons pass through, okay, for the bones it's x-ray. For the electron microscope, it's the electrons which go through. So the electron cannot pass through this pore region. I mean this mass here, which has biumbonate plug material, okay. Then in Basidiomycotina, the complicated type of pore with a single opening called as Dholiopore septum. Okay, so this one is the Dholiopore. Okay, it looks very complicated. It has borders also. It has some sort of covering from up and from down. Okay, so it's a very complicated looking pore. Okay, in the which family was that? Which family? In the Basidiomycotina. Okay, these are your these are the higher fungus, no, that you can see with your naked eyes. Example, yeah, in the when you grow your crops, no, sometimes those of you live in village, you grow rice or you know, then you see some powdery something. Yeah, those are what belong to the fungus Basidiomycotina. Okay, these those are that cause disease in your crops. Anything powdery, you know, girls, if you see your plants, okay, I'm not talking about just food crops, but any plant, no, you see something powdery, you know, it's not dust, it's not dirt, it's fungus only, okay, it's fungus. But it's not dangerous, it's not human fungus, no, it's plant fungus, okay. It's not that it can jump from plant directly to human, no. It cannot jump like that. Animal fungus or animal virus, they can jump to us because we are sort of the same mammalian species, no? Like this coronavirus, they say it jumped from the bat, okay? But don't have to worry when you touch a plant, it's not going to jump because it cannot break the barrier, the species barrier. Like it has adapted itself to infect plant, it cannot suddenly like become like a like coronavirus. It cannot happen. Okay, there's species barrier. Okay, so I think the diagrams are okay. These are the small pores, then the single pore is simple pore, then this one is a trachete pore, no? This is a complicated Dholiopore. Then this is when there's no pore. So always remember to add diagrams, okay? I keep repeating the same thing again and again, but you don't know, girls, because when we correct, no? Like for me, I'm just talking about my own personal experience. First thing I notice is a diagram. If the girl or the student knows the diagram, then I'm lenient. Okay, I'm lenient giving marks. Okay, so you have to use these tricks. Even when you go to masters, no, you practice your diagram first. Then you have to keep practicing so that you can draw fast and you draw neat. Okay, even when you go for masters, you draw quickly. You know, then you write. Okay, write quickly. But when you have to draw, no, the best technique, the best way, there's no secret, it's practice. Once you practice the drawing, no, it will be very fast and you'll be neat. It's like that, no? The more you practice something, the more faster, the more neater you become. But if suddenly you just studied it and then you are going to draw in the exam, no, it will be untidy only, girls. You cannot help yourself, because when we make mistake and we down, it'll be untidy. Okay, I talk too much, no? Okay, let me continue. So the lowermost fungi means the type of fungi which is not as highly evolved as the other. They don't have any pores, okay? Like the unicellular ones, no, they don't have pores, okay? Now let's go to the cells, okay? The cells of the fungus have a distinct cell wall, except for slime molds, okay? Slime molds, no, they are slimy, no? They have like a weird shape. They don't have a proper shape because they don't have cell wall. Okay, but the rest of the fungus, they have a cell wall, and it's not made up of cellulose, no, it's made up of fungal cellulose. Okay, that is chitin. In the plant cells, no, you will say, Miss, is cellulose cell wall. Yes, you are very much correct, but in fungus, it is chitin cell wall. Okay, this word I'm circling right now is chitin cell wall. Okay, please don't confuse. We tend to just write cell wall is cellulose in fungus. No, you have to mention that is chitinous cell wall, or chitin, the fungal cellulose made up of chitin. Okay. Then fungal cells, they don't have chlorophyll, but they do have protoplasm, nucleus plus cytoplasm, which has vacuoles, and it contains many nuclei. It has other organelles like mitochondria, endoplasmic reticulum, ribosome, microbody, vesicles, etc. Your normal organelles, okay? You can write etc. means endoplasmic reticulum, Golgi body, whatnot, ribosome, then what else? Smooth endoplasmic reticulum, rough endoplasmic reticulum, all those, they have everything, okay? Then the chief reserve food is your glycogen and oil. Okay, so reserve food is glycogen and oil. So what about plants? What is reserve food? You will say, Miss, is glucose. But here in fungus, it's glycogen, okay? Similar to glucose, but a little bit different in structure. Okay, let's look at the next. Mycelium that contains only one nucleus is called monokaryotic mycelium, or primary mycelium. It's easy enough. One nucleus means monokaryotic mycelium. Then when they contain two nuclei, that is binucleate mycelium, N plus N, formed due to the transfer of nuclei from one monokaryotic mycelium to the other, and initially form dikaryotic cell, and then to dikaryotic mycelium. It's also known as secondary mycelium. Okay, now let me explain. I'll just draw out my finger again. So we start with this monokaryotic mycelium. Okay, here in this cell, there's one nuclei, this is N. In this cell, there is another nuclei, this is N. Now what happens if we, okay, this is N, this is N. If we, let's say we break down this cell wall, okay, let me just, okay, then, so this one is in the same cytoplasm, right? So for a short time, it becomes n plus n, because this is one nucleus, this is another nucleus, so it's n plus n. So when it's n plus n, it is a dikaryotic cell. Okay, let's go back to the way it's written. When they transfer the nuclei from one monokaryote to the other, they form a dikaryotic cell. Okay, means a cell which has two nuclei, n plus n. And then slowly, these nuclei, they tend to get attracted to each other and they will fuse. Okay, so when they fuse, something like that, no? It's all still n plus n, but almost. Then when they do fuse, they will become what? 2n. So I think that part is clear. I think so. If any doubt, you can always text me in the group. I can always explain again. But I think it's clear enough. Okay. But this stage is short-lived, okay? All right. They have a tendency to separate again. They prefer to live independent, n plus n. Means they marry, they get divorced. What not kind of examples, but it makes you be able to understand, okay? It makes you catch these things. Okay, so let's continue. In the lower fungus, the reproductive cells, the zoospore or gamete may be uniflagellate or biflagellate with a 9+2 organization. So now we are talking about the reproductive cells, okay? Now best is just to go through, look at the image. So here, this one is a whiplash type which doesn't have hairs in this side, it's just a plain, okay? Then this one is a uniflagellate, but it has tinsel, it has the hairs at the side. Then these are your biflagellate. They could be coming from the tip, they could be coming from the middle, or they could be coming biflagellate, but in a whiplash type. Okay, so again, please draw the diagram. Now this one, remember when we talked about the cell? Okay, just look. Remember, we have a cell wall. I better make it smaller, no? Okay, so we when you start to draw, first you drew like this, no? You drew a cell wall. Okay, fine, we are done. Then you draw a cell membrane. Okay, we are done. Then you drew the cytoplasm. You gave ... Then you drew a nucleus. Okay, nucleus with a nuclear membrane with some pores in between. Okay, so you drew the nucleus and you drew the nucleolus. Okay, nucleolus is inside the nucleus. It's like a cell is big, then a nucleus, then a nucleolus. Okay. Then you drew the mitochondria, you drew the endoplasmic reticulum. No, this long one. Okay, endoplasmic reticulum. Then you drew the ... ribosomes. Okay. So as you draw your lomazome, also there's something different known as lomazome. So again, the only way to study diagram is to practice them. Okay, so you just draw them by hand. When you keep drawing, no, when you start from the outermost, you start from the cell wall, then the cell membrane, then you go cytoplasm, then nuclear envelope, then you draw nuclear pores, the nucleolus, then you go organelles. It will come to you. You'll be fast and you'll be neat. Okay, so when you look, when we cut, okay, when it's almost half an hour. Okay, so if you cut the TS of this flagella, no, if you cut it and you look in an electron microscope, okay, you look through a TS, you will see that structure is like this. You will see a 9+2 arrangement of microtubules. Means this is 1, this is 2, 3, 4, 5, 6, 7, 8, 9. Okay, 9 microtubules that are together. Okay, in double 2 and then plus 2. Okay, 1 and 2. You get it? So this is 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, and 1, 2. So it's a 9 plus 2 arrangement. Okay, and this arrangement, no, is everywhere. It's in my muscles, your muscles. Okay, it's in the what to say, all those microtubules, no, that is there in the higher plants also. This is how nature has devised itself, how to make the stick strong. Like remember that a moral of a story, no? Like remember, like some brothers, there were 10 brothers, no? They were fighting among themselves. Then the father got angry and he took one stick, okay? He grabbed 10 sticks and he started breaking one by one. Then he said, if you are alone, you will break easily. Then when he grabbed 10 sticks together, he could not break, no? So that is how nature itself has devised, okay? If it arranges itself in this particular nature, this 9 plus 2 arrangement, nothing can break, no matter how much this gamete is swimming, no? This zoospore swim from, ocean to ocean, river to river, no? It will not break because God has, nature has, God has arranged in such a way that they will be strong, okay? So again, you have to draw. These are the arms, the microtubule arms. That's the central fiber. Those are the sheaths and then the secondary fiber. Okay, let's look at what they've written. The zoospores may be uniflagellate or biflagellate with a 9 plus 2 arrangement. They might be whiplash or smooth-walled. Okay. They may be covered with a small hair-like known as mastigomias or filaments or tinsel, and they originate from the axil filament. Okay, the tinsel type gives forward movement and the whiplash type gives beating movement. Okay. So like this one has both. No, so this one will go forward and this one will beat. This one will go forward, this one will beat. Okay, this one will only beat. This one will both of them will beat. Okay, this one will move forward. So with this chapter is done. Thank you.